Thank you very much, Nick. And I can see that my microphone works. That's good. At the back, can everyone hear me okay? And is that good? You're more used to this lecture theatre than I am. I love all this space. It's fantastic. It's almost like I'm going to start some interpretive dance or something like that. So if I get too enthusiastic, stop me. I also feel the need to sort of feel presidential and sort of say, how are you? Right. So what am I going to talk to you about? I'm going to talk to you about some stuff I do in my day job, working on the neural basis of awareness. So my fundamental interest as a cognitive neuroscientist is really in why people do the things they do, why people behave the way you're behaving right now. And of course, if you think about the phenotype of humans as opposed to any other animal, we've got a number of distinctive characteristics. For example, we gather in large groups like this, where we're all after the age of sexual maturity, but we're still engaging in teaching and learning, that extended thing. We have hospitals where we care for people who are genetically unrelated to us, and if we see somebody who's hit in the road, we'd stop and help them, even though they are no genetic relationship to us. We have language with which we communicate in a flexible and extendable fashion, and so on and so forth. But we're also aware and self-aware. Now, any one of those, and there are other characteristics of the human phenotype, you could argue about whether they are unique to humans or not. But collectively, they form a unique phenotype. And that is why, although we share a lot of our genome with mice, of course, mice do not have universities, lectures, hospitals, and other things like that. They scuttle around in the floorboards. There's something special about that. So I'm going to talk to you a little bit about that, and I'm going to focus on the area I work on most, which is vision. The standard approach to that is illustrated here in this slide from my postdoctoral advisor, Christoph Koch. So it says that things in the outside world produce activity in our brain, and that activity in our brain is in different types of cell, different types of nerve cell, and has different patterns of firing. And some of it, the NCC or neural correlate of consciousness, is associated with a particular concept, a conscious percept we have, in this case of a dog. So there's a relationship between what we perceive, what we think we see, and some aspect of firing or structural anatomy in the brain. The task of the cognitive neuroscientist is therefore to try and find out what that NCC is and what commonalities underpin all our ability to have those visual experiences. No sign of a pointer, never mind. What I'm going to talk to you about, though, is something a little bit different. It's about individual differences. So there are these two complementary methods in psychology, one of which is aimed at this kind of a question, which is how do we find the commonalities that underpin all our ability to see things. But we can also learn something about those by studying the differences in how we see the world. Now, you might think that's a bit of an odd question to ask, because you think, well, we can all see the world. What about the differences? But I'm going to show you some of those. Let's start, though, with a little bit about the brain. You don't need to know a lot about the brain for this lecture, but here what I'm showing you are three left hemispheres, the back bit of the brain, and the occipital lobe, where the visual cortex or the visual brain lives. These are three hemispheres from three people, and highlighted in purple there is primary visual cortex, or V1. And we can find out where that is by using functional magnetic resonance imaging to delineate that area. And I'll talk a bit about that later. But I want you to notice a few things about that slide. This is where a pointer is going to come in handy, but we don't have one. Right. You'll just have to imagine. If you've got a big stick, that's good. If you've got a pointer, that's good. If you look at those three hemispheres, you can see they're all about the same size. You can see the sulcal and gyral pattern is slightly different in each individual. But in each individual, you can see a sulcus extending from the very back, there, anteriorly, right, the wiggly one that goes through the purple bit, and it's the same there, although it's a slightly different shape, and it's the same there, but it's a slightly different shape. That's because primary visual cortex is always found in the calcarine sulcus, which is the name that neuroanatomists give that particular valley or sulcus. Now, you can also see by eye, though, that there's quite a lot of variability in the overall size of that purple area. So the person in the middle, AAB, appears to have quite a lot of purple, even though their overall brain doesn't vary much in size. Whereas ARW, even accounting for the folding as a sort of medium-sized bit, and RFD has a sort of small purple bit. So by eye, there seems to be quite a lot of variability in how big visual cortex is. Nick is squatting down here, desperately trying to find one. But it's okay, we're doing well. Now, That was non-invasive brain imaging. If you're lucky enough to have a whole load of human brains like Tim Andrews in York had, you can do post-mortem studies of those same structures. And that's what's shown here. So each point is a single dead human brain. And the purple area I was talking about in the previous slide is on this axis here, V1 area. The other axes are looking at the size or the surface area of other bits early in the visual system on the way to that primary visual cortex, the optic tract and the lateral geniculate nucleus. It doesn't particularly matter knowing about where those are. What does matter is that you notice that all three of those measures are correlated. So it seems that if you've got a big primary visual cortex, thank you very much, that's very, very kind. If you've got a big primary visual cortex, then you've got a big rest of your early visual system, and if you've got a small primary visual cortex, you've got a small rest of your visual system. So something is scaling up and down the size of the early bits of our brain to do with vision. Those of you sitting at the front can also see the size of this effect. So those who can't see, here is about 1,500 square meters, here is about 3,500 square millimeters. That's a variation of about 3. So that's saying in an audience like this, if I could measure your V1s by killing you and post-mortem looking at your brains, which would be ethically unacceptable, I would find a variability of a factor of about 3 in the size of your visual cortex. I don't know if that surprises you, but it surprises me a lot, because I tend to think of individual variability as not being something fundamental like vision, but something perhaps to do with our preferences or experiences. You know, you like Marla, I like Lady Gaga, that kind of thing, something in the frontal lobes. But here we have at the earliest stages of vision really quite substantial variability. It's not variability in brain size, because variability in brain size is about 20%. People's heads don't differ that much in size, and by the way, difference in size of head doesn't actually correlate with difference in size of brains. So you have a big head, small brain, or small head, big brain. So, what are the consequences of that? I was interested in asking the question, does that make any difference to how we see the world? And this is Sam Schwarzkopf and Chen Song who did the work I'm going to talk about now. We started by thinking, well, if you took these two people, AAB, who's got a big V1, and RFD, who's got a small V1, what are the consequences potentially for how that visual cortex works? Visual cortex works very simply. This bit of the brain, the left visual brain, looks at the right-hand half of the visual world. So if I asked you to look at that point there, the left-hand side of your visual cortex would be representing the right visual field. And let's imagine I shone a couple of lights, a red light and a blue light at those positions in the visual field. What we know about early visual cortex, we've known for over 100 years, is it forms a sort of map of the visual world, where there's a relationship between where you stimulate in the outside world and where in that map gets activated, neurons that get activated. If we unfolded that, the map roughly looks like an oval like this, where the center of vision, the fovea, is represented at the back, and the periphery of vision, out here, is represented at the front. And then rather confusingly, it's upside down, so the lower bit of the visual field is represented above, and the upper bit of the visual field is represented below. So putting all that together, we can predict that in RFD, the red and blue lights would produce, well, not colored activations, because brain activations don't have any color, but they would produce activity in those two bits of that visual map. Now, what about the other person, AAB? Well, they've got a bigger V1, so quite literally, they have a bigger map, just physically bigger. And so, while the locations in the map are exactly corresponding in terms of where they are in the map, the distance they are apart physically on the cortical surface is greater. Now, that's the only complicated bit of this lecture, because if you followed that, what that's saying is it enables us to make a prediction. The prediction is, if there's any property of vision that depends on this blue thing being influenced by something like the red thing, something somewhere else in the visual field, what you're looking at gets influenced by something over here. In a small map, those influences should be stronger because everything's closer together, and in a big map, they should be weaker. So to test that hypothesis, all we need to do is find an example of that. And we scratched our heads for a while, and then we came up with this example. This is the Ebbinghaus illusion. Things, in this case the orange disk here, are influenced by things over here. What do you see when you look at this? You see two orange disks surrounded by some grey inducers, and I guarantee that every single person in this audience sees that disk as larger than the other disk. It is, of course, an illusion, because if we remove the inducers, they're physically identical. And if we bring the inducers back, the dissimilarity appears. So that's an example of where two physically identical objects can appear perceptually dissimilar. And why does that happen? Well, there's lots of theories, and I could give a whole rather boring undergraduate psychology lecture about it, but broadly speaking, this stuff is somehow influencing this stuff in the brain. Now remember my map prediction. If stuff is further apart, that influence should be weaker. So the prediction is, in people with a big V1, they should see a weaker illusion. What do I mean by weaker? I mean, we need some way of measuring how strong this difference is in each of you. Now, if you imagine I have a little knob, and I turn the knob, you can imagine I could make this bigger and bigger. And if I give that knob to you, you could make it bigger and bigger until the two orange discs appear identical in size. That physical offset you've added to the left-hand orange disc would be a measure of how strong it would be. And in essence, that's what we devised experimentally. Now, I can't reproduce that for each of you in a lecture, but what Sam has done here is set up a few examples, and we'll test you on this. Okay? So round the outside, all of these have a slightly different physical offset, as if you turn the knob to a slightly different location. For all of you, I want you to look at which one of those matches the one in the middle. Okay? For each of you, think what it's going to be. And I'm going to ask you in a moment to put your hands up to indicate what your match is. Don't stress out over it. It's visual psychophysics, okay? It's what it looks like. There isn't a right answer. Well, actually, technically, there is a right answer, but you're not going to be tested on it. Right, everyone got their match? Right, this only works if you all participate. So, who matches the 12 o'clock one? Hooray, and I'll tell you why in a second. Who matches that one? Who matches the 3 o'clock one? Starting to get some people. Who matches the 5 o'clock? Notice your audience. A few more. Who matches the 6 o'clock? About the same. Who matches this one? Quite a lot. Who matches the 9 o'clock? Just a few. And who matches round here at whatever it is, 10 o'clock? Maybe one or two. Right. Existence proof. You all saw the illusion, and you all saw a different strength of that visual illusion. And it was, roughly speaking, a Gaussian centered around here. How do I know you all saw the illusion? Because I told you there was a right answer. The veridical match is at the top, the one that nobody saw. So everyone saw the illusion, but, thank you very much, even better. Fantastic. It's the same. That's good. But I saved somebody's battery, which is good. Thank you so much. Okay, so as you were, you all matched a slightly different strength. So that suggests that all of you see the illusion with a slightly different strength. And what I can't show you is if I came back tomorrow and asked you for that preference would be stable over time. So it represents a trait, if you like, a trait that you have in you. Now, our prediction was quite straightforward, if you remember. The strength of that illusion would relate to the size of visual cortex, and weaker illusions would be associated with stronger visual cortices, with bigger visual cortices. And so we tested that for the Ebbinghaus illusion, and also for another illusion which is based on three-dimensional context, where these two again appear dissimilar sizes. Different mechanism, but it's going to turn out to be the same result. Here's the result. The graphs are very simple. Each point is a person, and this is the area of their visual cortex that we measure with functional MRI, and this is the strength of the illusion. A weaker illusion, as you can see, is associated with a bigger visual cortex. Now, that doesn't prove that that mechanism is correct, but that certainly is consistent with that hypothesis that these lateral interactions in visual cortex mediate how you see the world, and that how you see the world, at least in terms of size, varies quite substantially across people. That's actually just specific to that visual area, because these other visual areas I haven't talked about, the other colors in that very first slide I showed you, don't show that relationship. So it's something special about the very earliest visual area. And that relationship I just showed you is shared by other illusions. So this illusion is a well-known visual illusion called the tilt illusion. Those two central patches, this one and this one, have the same orientation. But for all of you, the one on the left appears pulled round slightly anti-clockwise, and for me, I should say, I see the illusion too. And the reason for that is this context here, which pulls round the perception. Now again, we don't need to worry about the mechanism, but it shows the same relationship with V1 surface area. And for those who are really interested and want a sort of extra super duper master's level piece of information. We also see that relationship just for the discrimination of orientation differences. So the just noticeable difference between discriminating a tiny change in orientation is also correlated. And for those who are really interested in that, the nerdy additional fact to do with that is recall that probably orientation tuning in early visual cortex is a property of lateral connectivity in visual cortex. For those who are not nerdy, don't worry about that particular statement. Stay with the main story. So what are we doing now? We've shown there that there's a systematic relationship between the anatomy of early visual cortex and how you see the world. And what we're trying to do now, that I'm not going to go into endless detail about, is we're trying to understand how that relates to the architecture of visual cortex. Because we know a lot about how visual cortex is structured from experiments in animals. And those experiments allow us to build simple computational models that help us try and test whether what I just said is true. And for those who are really interested, you can go and have a look at Neuron this month. Neuron has an open access article, so you can all download it, with Chen Song, who did the work in this paper, where she looks in a lot more detail, as this slide illustrates, looking in detail at the structure of early visual cortex, and looking in particular at the relationship between cortical thickness, local surface area, and some of these elementary visual properties. But that's a little more detail if you're interested. Let's move on. So why are we doing all this? This is anatomy. We're also interested in understanding how visual cortex functions. And so let me show you just one experiment there. We saw this variability in early visual cortex size. Does that have any consequences for brain activity? All we've done so far is measure how people see the world and the anatomy. What about measuring brain function? So just one experiment to illustrate this that Sam Schwarzkopf did a couple of years ago. This is in a different type of scanner called a MEG, or magnetoencephalographic scanner. This is an experiment where we measured something called induced gamma oscillations, which sound complicated, and this slide looks complicated, but in fact they're very simple. If I measure the electrical or the magnetic activity associated with that in an MEG scanner from your visual cortex, while I flash a stimulus in your visual field, I will see after I show that stimulus, there's a burst of activity. Funnily enough. What I can do is measure the different frequencies that are contained in that activity, a bit like in a hi-fi you can get the bass and the treble and the different frequencies separated out and measure their power. If I do that, when I flash the stimulus at time 0, the burst of activity I'm talking about is illustrated by the heat map here, showing there's kind of different frequencies that become hot at different times over the first 1.5 seconds. As I flash that stimulus, there's a burst of activity and then it decays away. This one is particularly interesting because it's in this frequency range up here, which is known as gamma. You may have heard of gamma oscillations or been taught about those in your undergraduate neurophysiology and psychology. So this is just a standard boring response of how everyone's visual brain responds. But there's an interesting individual difference here. And that individual difference is with the peak of where that heat map is. We can see that a bit more clearly if we show the same subject in a different way, where we're mapping the power, which is like the strength of this yellowness, as a function of frequency during that first second, this second here. When there's no stimulus present, this just shows what's called a 1/f spectrum, but when the stimulus is there, this peak here is corresponding to this hot spot there. And it enables us to see much more clearly that that's at or around 40 hertz in this person. And I say in this person because, of course, this is just another person. It looks qualitatively quite similar. There's, again, a peak, but you can see by eye it's higher. And indeed, if you plot it, it's actually a bit more jagged and rises to a peak here. It's about 50 hertz. That's been known for a while. That is, if you flash the stimulus and measure that response in each of you, there will be a different peak gamma in every single one of you. Why is that? Well, we thought that might be something to do with, guess what? The surface area of visual cortex. And the reason we thought that was because there are theoretical models of how that induced gamma comes about, which rely on different neurons interacting, again, guess what? Laterally in cortex. And so we made a very simple prediction that if we tested the peak gamma in a load of people and then plotted it as a function of the area of visual cortex, we get a nice relationship, and indeed we did, as you can see. So there's a nice linkage there, I think, between anatomy, function, and how people see the world. Why might we want to do all that? Well, lots of reasons. I'm interested in just how the brain works and why people do the things they do. But some people are also interested in some of these visual illusions in particular situations. And one I've illustrated here is one Steve Dakin and Uta Frith highlighted a number of years ago, which is people with autism spectrum disorder seem to have a relative insensitivity to global context. Now, again, we could do a whole lecture on that, but let's concentrate on what that means in the context of the illusion I explained. You all saw that Ebbinghaus illusion because of the global context influencing what the orange discs look like. But if you have autism spectrum disorder, many individuals do not see that illusion, either as strongly or at all, because they are relatively insensitive to the global context. And there are a number of theories about why that should be. Our theory, of course, is that there might be something not quite right with the architecture of their visual cortex, because in healthy individuals, I've just shown you lots of data to suggest there's a relationship between what the visual cortex is like in healthy individuals and this particular illusion. And so what we've done so far is we've done a very preliminary investigation, or Sam has, I should say, of what visual cortex looks like in autism, because funnily enough, nobody's done that. The way we do it is rather complicated, but I can walk it through very simply. This looks very confusing. In fact, it's a brain pointing at you, with the occipital lobe pointing at you. And what we've done is use functional MRI to do something a little more complicated than the purple area I showed you in the first slide. We've actually mapped that purple area and its fellows by moving a stimulus across the visual field, where the color code now is corresponding to stimulation in different parts of the visual field. So this part really likes the right visual field because it's left hemisphere, and the reddy bits and the greeny bits are at the top and bottom respectively. You can see on this map here, the red bits corresponding to the central visual field are sort of here, and then as we go out, it gets more yellowy and bluey. That's called retinotopic mapping. It's because there's a map in visual cortex. If we move a stimulus across your visual field, we can get activations that stimulate different parts of your brain in that map. Now, what we can also do is be a bit more sophisticated, because just as each point in this image is a pixel, because it's a slide projected through PowerPoint, each three-dimensional bit of the brain is a voxel. A voxel is to a volume as a pixel is to a picture. And for each voxel, we can actually characterize the precise bit of the visual field that it responds to, both in terms of its location. It might be a small bit of the visual field here in the upper left quadrant, or it might be a larger bit of the visual field here in the center. So we can get a number of parameters out of these maps which say how big, for each location in the visual brain, where is it in the visual field that it responds to, and how big is that? Now, if you followed that, then you'll realize we can get another type of map. So here's some data, okay, from autistic individuals and controls, healthy controls. Slightly different format. Now the brains are globular, just to confuse you even more. That's fine too, because this is the visual brain that's centered here. We've blown up the brain and made it look like a beach ball, rotated it to the visual brain is right in front of you, and painted on the activity from functional MRI. You can see these maps of phase angle and eccentricity here. So for example here, look at this, in a healthy control, the very center of the visual brain is responding to the very center of the visual field, and as we go out, we respond to more peripheral bits of the visual field. This is a map, though, of how big those receptive fields are, right? Are they really, really small? Really, really big, like the central visual field one. And you can see, in general, they're quite small in V1, and they get bigger in these other visual areas. Now, what we're really interested in doing is saying, is there a big difference between controls and autism? Is there some difference in the architecture? By eye, you can have a look at those, and they look macroscopically pretty much the same. And that's true. They are macroscopically much the same. To see the abnormality, you have to plot it out in a bit more detail. And I'm going to concentrate just on these two graphs for reasons of time. What's being plotted here is eccentricity. So where, compared to the center of these maps are we, or compared to the center of vision? As you go out, okay? 0 is at the center. Positive values are further out. How big are the receptive fields? Small or big? What you can find, and this is plotted for a whole load of visual areas, but it's going to turn out to be roughly the same result. So let's concentrate on, let's concentrate on this pair here. Actually, or this pair here, that's probably easier because it's further up. These are from V3, okay, the third visual area. What you can see is as we go out in eccentricity, as we go out in the visual field, the receptor fields get bigger and bigger. That's normal, and that's known, and that's predicted from what we know in animals, that out here in peripheral vision, your vision is a bit more blurry, your receptor fields are a bit bigger, and down here it's a lot more detailed. But what we didn't expect is the difference between this yellow line and this blue line. Autism versus control. They're quite clearly different in size at all eccentricities. And that's also true for V2 here. It doesn't seem to be true, interestingly, for V1, and it's true here for MT, the visual motion area, but not so much true for V4 or V3. So that's preliminary data, but that's data that suggests there's something not quite right about the visual cortical structure in these individuals with autism. They're all adults living in the community. They all have this relative insensitivity to global context. And what we've got here is some systematic difference between them and age-matched controls. So what we're doing now is trying to apply our methodology to do with illusions to these individuals to say, can we think about this visual abnormality in autism in the context of our theory? Okay, so I think I've said quite enough about what's been discovered, and I'm going to talk about two different structures for the remaining 15 minutes or so, okay? Because I want to give you a bit of variety. I've talked about primary visual cortex a lot, V1. I'm going to talk now about parietal cortex and prefrontal cortex and give you just a flavor of their role in vision and their individual variability. So let's start off with parietal cortex. This is work Ryota Kanai with Bahadur Barami, David Carmel, and Megumi Fukuda did. Right, I like parietal cortex. I've worked on it for ages. I've worked on it for ages because it seems to be involved in all of these illusions. The one at the top is the most difficult one to show because it requires a mirror stereoscope. Funnily enough, we don't have one of those in this lecture theatre. This is called binocular rivalry and occurs when you show two different images to each eye, in this case, a grating and a face. And what I can't show you, but you can experience if you go and see your nearest friendly mirror stereoscope guy or gal, is that your perception alternates spontaneously between each monocular view. Actually, I can do a demo. I can do a demo. Let's do a demo of binocular competition. You are students, right? So you must have an A4 piece of paper, correct? Good. Take an A4 piece of paper and roll it up to form a little telescope. Psychologists in the audience, humour me. Yeah, I'll take an extra 2 minutes, but it's fine. This is good. I like this one. Right, make a telescope, a longitudinal telescope. I should demo so you get the right thing. I haven't got the right. Right, here's my times higher for all you jaundiced academics. Right, so you're making something like this. Right, settle down, everyone. Okay? Right, hold it firmly, but securely, not crushing it, in your hand. Your other hand, what I want you to do is don't place it over the end. This is a test of your final degree grade. But at the end, no, not here, further down the tube. Okay, so it's like that, right? One hand at one end, the palm at the other end, all right? Everyone got their apparatus assembled. Right, what you're going to do is you're going to put it to your eyes, so one eye is looking down the tube and the other eye is staring at your palm. Okay? Got to get it to the correct eye, okay? Like this. Do it, do it, do it. Now, look at me. And if you've got it all configured correctly, you should be looking at me through a hole in your hand. Good. The titter of laughter suggests that that experiment has worked. Right. Okay. Settle down, everyone. So, existence proof of binocular competition. So what was happening there? Okay, so we concocted a situation artificially where for a particular part of your visual field, one eye was seeing my head, and the other eye was seeing a rather boring 10 pp sized piece of your palm. What you experienced was in that particular location in the visual field, you did not see a mixture of the two. Yeah, it's a mixture of the whole thing, holding your hand and all that stuff, but in that location, it wasn't a mixture. There must have been some competition, and my head must have won out because you saw my head rather than that palm of your hand. Even though the most consistent thing to see, notice, was actually to see your palm of your hand, you saw my head in that hole. Now, if we optimize the conditions more, you would actually see an oscillation between those two. You don't see an oscillation because of, depth and all sorts of things like that. But that's good. And have fun at home with that illusion. Now, that illusion is one of a family of what are called bistable illusions by psychologists. I'm sure many of you are familiar with the Rubin face vase here, where this contour is ambiguous. and can be a nose, a mouth of the Queen and Prince Philip looking at each other, or it can be a vase. But it can't be both at the same time, and it oscillates between the two. The Necker cube here experiences perspective reversals between those two disambiguated versions. In other words, this can be down and to the left, or up and to the right. The cube can go up and to the right. All of those illusions share some common properties. Alternations happen spontaneously, and those alternations activate these areas of frontal and parietal cortex, which is interesting. But what's more interesting to me is that all of these share quite considerable individual differences. So let's try one last demo, okay? This is good for people at the back, where I always used to sit during my undergraduate lectures, because you're going to be able to see your colleagues in the front. So this is a structure from motion stimulus, a pretty bad one for the psychologists amongst you, but it should be rotating in one direction, and if you fixate on the red dot, every so often, and if it doesn't, you can flick your eyes just to demo, it should swap directions and rotate in the opposite direction. Okay, so that's the alternation, that's the bistability. What I want you to do is indicate to your fellow lecture attendees your alternations using your arm like a paddle, please. Slightly embarrassing, but please do it. Okay, those of you at the back, look at the variation in switch rate across the audience. Okay, so what we're seeing by eye, switch rate inversely correlated with IQ. That's a joke. What we can see by eye is some people are switching quite quickly, some people are stuck in one configuration they can't get out of, story of my life, and other people are at intermediate levels. Okay, that's enough, right. So that's a slightly harder demo to do, and of course, those amongst you who are psychologists will be moaning that we didn't control fixation and so on. It's not intended for that, right? It's a demo. If you actually measure that as Ryota did, and you measure it here in a bunch of 30 people, but it would be the same result if we measured it in a lecture theatre of 150, you get this sort of distribution. This is the number of people, and this is the average time between those switches. So what I want you to notice, there's a distribution there, okay? Lots of people around here, not many people out here, but look at this difference in time, okay? Some people are switching on average every 2 seconds, whereas some people are switching on average every 14 seconds. 7-fold difference. Okay, so there's a huge difference, almost an order of magnitude difference across people in this lecture theatre in how fast they switch. Why is that then? Well, it turns out there aren't really any very good theories about that. There are some psychologists who theorize it's to do with how manic you are, right? You know, you're more excitable and manic, you switch faster. And if you're less, more depressed, you switch slower. That's not a great theory, and there's not a lot of empirical support for that. And we didn't know what the answer was, but we had a very simple theory, which was perhaps that's something to do with the parietal cortex that activates when the switches happen. And perhaps there's something different about the structure of parietal cortex that correlates with that switch rate. And so we did a really simple experiment. We took a bunch of structural brain scans of people like you, measured the switch rate, and then throughout the whole brain said, is there anywhere in the brain where this distribution of switch rates correlates with the thickness of the grey matter in cortex? Previously, we were looking at surface area of V1, but now we're just looking at the whole brain. And the answer, it turns out, are these regions here. I'm going to concentrate on the posterior region because that's the one that replicates, and that's shown here in a different type of analyses. These are two different packages called FreeSurfer and SPM, giving the same result. But then again, it's the same data, so perhaps you're not so surprised about that. These regions here show an inverse correlation, that's why it's in blue, between switch rate and local grey matter volume or cortical thickness. So those areas of parietal cortex that activate when your switches happen, the overall thickness of them in you or I predicts your switch rate. You might say, it's just a correlation. So what Ryota did was he went out and applied transcranial magnetic stimulation to those regions. So this is a technique that transiently disrupts that region of the brain. And when we stimulate over that left SPL for superior parietal lobule, or right superior parietal lobule, before and after, and measure that time between switches, in all the subjects it goes, well, pretty much all of them, but certainly in the group, it goes up. But it doesn't for a control region over the vertex, just the top of the head, which we don't think is involved. So that region, whose structure varies and predicts how fast you switch, when we knock it out, it alters switch rate. So it's causally involved in that. So that's, I hope, a nice demonstration of how we can apply this individual differences methodology a little more generally to variability now of bistable perception. So in the last bit of the lecture, I want to move to prefrontal cortex. And this is some work that Ramona Weil and Steve Fleming here did with me a couple of years ago, just over a couple of years ago. So what does the prefrontal cortex do in vision? It does a lot of things for cognitive functioning generally. In the domain of vision, the thing I'm going to be particularly interested in here is metacognition. Okay, now, metacognition is a big area. It's thinking about thinking. God, how profound that is. What does that actually mean? Well, let's try and bottom it out. You can all introspect about your visual experiences. Okay, so let's imagine a transient visual experience here happens. The lecture theatre door bursts open, right? And you catch a very brief glimpse of a partially clothed individual. Okay? And you think that looks a little like your mother. Right? How horrific, okay? Ultimate embarrassment. Partially clothed mother appears in lecture theatre at university. But the door bangs shut again. That's gone. How certain are you that that was your mother? Because that's a really important question to ask right now, right? You don't know whether you're right or wrong. The lecturer is about to go and investigate, but it could be critical to you to work out how certain you are about that glimpse you just caught out of the corner of your eye. So what we've got there is a situation where there is a correct or a wrong answer, right, to that visual discrimination you just made on the basis of incomplete information. Might be your mother, might not. We will find out shortly. But before you get that feedback, you can make a judgment by introspecting, and you can say, well, I got a really clear view, it's definitely my mum, I'm out of here, that's terrible, right? Or you can say, no, definitely didn't look like her, I got a clear view. Or you might be more uncertain, you might say, well, I was concentrating on the lecturer because he was giving me such a great lecture, I just caught it out the corner of my eye, I'm really uncertain, this is really worrying me. So you can make a judgment about your confidence based on introspection about your experience. That is the form of metacognition that I'm going to talk about now in association with the frontal lobe. Okay? There are other forms of metacognition. For example, when you're revising for your exams, you know when you know something. You know when to stop, hopefully, as well, in terms of revising. So you have some introspective access to meta-memory. And there are other forms of metacognition. But I'm talking here about perceptual. So how do we operationalize that to do an experiment on it? Well, very simply. We do a very boring visual experiment, having talked in interesting terms about partially clothed mothers. Now this is how it actually turns out in the psychology laboratory. Two experiments, but it's the same result for each, so I'll just take you through this one. Imagine you're fixating here, and we flash these gratings around fixation. There's a bunch of them, and one of them, as you can see, is higher in contrast. Then they go off the screen, and we flash a second array of stimuli, of those gratings, again around fixation, and as you can see, they're all the same contrast. The visual discrimination we want you to make in that two alternative forced choice task is to tell us a very simple thing. Was it the first or second interval that contained that pop-out high contrast stimulus? So it's a dead simple task. But before we tell you whether you were right or wrong, we also ask you to rate your confidence from 1 to 6. And obviously, in the example I've given you, it sounds very easy and like you're always going to be high confidence. So what we start to do is we start to make it harder and harder. We flash the arrays for relatively short periods of time, and we can turn down the contrast here until it gets harder and harder. And we turn down the contrast until all the people are performing at about 70% accurate. And that's going to be important because we want to rule out differences in discrimination accuracy. We then see if their confidence links to their accuracy. Sometimes they get it right, sometimes they get it wrong. On average, they're getting 7 out of 10 right. But how well do their confidence judgments link to their accuracy? How we do that is using something called signal detection theory. You are saved, because of the lateness of the hour, from me explaining anything about signal detection theory, which is either the most interesting thing you've ever heard, green and sweats, or the most boring thing you've ever heard. But broadly speaking, the idea here is we just want to say, not whether people are always confident, those are often called men sometimes, that's not important in the context of this experiment. We're not after people who are just relentlessly confident in a kind of tedious and boring way. We're after people who can modulate their confidence, so when they're going to get it right, they're generally confident. When they're maybe going to get it wrong, they're less confident. And people who can link like that, we derive a measure which we plot here. We call it the area under the curve. These are the data, and I've got one more data slide after this. Everyone is performing at 7 out of 10 correct. I told you that was deliberate because Steve manipulated the experiment to make sure that was the case. What we found, though, was across those individuals, the individuals varied really substantially in their ability to link their confidence to their performance. Some people were really good, and some people were really not very good. So again, there are really big individual differences in how we're able to introspect about our visual performance. But we could go a step further because now we can link that all the way back to the brain by asking where in the brain is there a correlation. And we find these areas in the front of the brain, Brodmann area 10, these areas don't replicate, and this area in precuneus that show a relationship between that parameter we measure and the grey matter volume. And that isn't seen, cognitive neuroscientists, for discrimination, for performance, a measure of performance. Okay, so skipping ahead. What I've shown you there is in prefrontal cortex, we can again show a similar linkage between individual differences and the structure of the brain. I'm going to wrap up now. Stuff we might like to come back to is where I show a linkage between V1 and the volume of this area. So I'll just perhaps show you this slide and then finish. Linking right back to the start of the lecture, I showed you that people's visual cortex varies a lot in size, but I told you that although that varied 3 times, differences in overall brain volume are about 20%. So the question is, what gives? If you've got a big visual cortex and the whole of the rest of your brain hasn't scaled up by the same size, what gets smaller? And the answer are these areas of prefrontal cortex, which are exactly or very closely the same to the areas I just showed you that are to do with introspection. So there's a linkage from the last thing I showed you in the lecture, difference in prefrontal cortex relates to metacognition, and the very first thing I showed you in the lecture. And that relationship is at the level of brain structure. Okay, so I'm going to wrap up now. What I hope you got from this lecture is that we can look at individual differences in the structure of our brains, the adult brain. And although that structure is constantly changing, after all, the reason I'm here now is to change the structure of your brain, just like all your lecturers. If we didn't, we haven't really succeeded because you haven't remembered anything. The structure of the brain also seems to encode quite a lot of individual variability that tells us something about complex human behaviors. In this case, how we see the world, how we introspect about the world, and how bistable perception works. I've shown you, in amidst all the other evidence, the way in which, as you know from cognitive neuroscience, we combine different methodologies. And the main part of the lecture was to link these 3 structures with the idea that I hope is now embedded, that although we can all see the world, our exact perceptual experience shows quite substantial individual differences across individuals, and by studying that, we can learn something new about the brain and how we do and we see the world the way we do. Thank you very much.
Anyone have a question?
Hi, thanks for that. I'm interested in the developmental story, particularly for autism.
Yep,
So,
go for it.
is it the differences in the brain to begin with that are leading to these differences in perception? Or is there something about how children, say with autism, are interacting with the world that gives them a different visual experience of the world that could then affect the way that the visual cortex develops?
So very good question, quite clearly, and thank you for introducing that. Correlations are not causation. So naturally, we can't tell the answer to that. We could find that out, but to find that out, we'd have to conduct a longitudinal study where we study the emergence of those properties over time and whether those properties were present very early on. It's quite challenging to do retinotopic mapping in children. The subjects I showed you were all adults, and I spend most of my life studying adults. I have some small children. I have, in fact, a small child who has been in a longitudinal MRI project, and believe you me, it's quite challenging to get her to stay still just for the structural scans. So it's an important question to resolve, and the short answer is we don't know. I mean, it's true, of course, for a whole load of developmental properties in autism and autism spectrum disorders, like theory of mind, that although they are delayed in their ability to do theory of mind, clearly some individuals with ASD learn strategies that are different to healthy neurotypical individuals that enable them to perform the task. So we can perform a task in two different ways. That said, I think theory of mind is a little bit different perhaps to early visual cortical properties. I think your ability to learn different strategies for how to structure visual cortex, for example, or how visual cortex works, is probably more limited than for those more complex behaviors. Interesting question, though. Am I directing this, or are you doing it, Nick? Go for it.
Kind of linking on that, my question was similar, although not related to autism, and it would be to what extent situational factors contribute to inter-individual differences in these areas, because we know from people who work on perception that illusions of depth, for example, are shaped by the environment people grew up in.
Yeah. Goldsmiths people.
So, yeah. So whether, for example, you grew up in a rainforest or on the plains will shape to what extent you create illusions of length.
Yes, for those who are a little confused about what that question was all about and why I suddenly said goldsmiths, this is referring to a famous set of studies. I mean, there are others, but ones that look at illusion I didn't talk about, called the Muller-Lyer illusion. So that's the illusion that many of you will see, which is if you have a line with two arrowheads at the end and a line with two arrowheads the other way around. Yeah, people are nodding, they've seen that. They appear to be different size. Now, there is work in the literature that suggests that different cultures perceive the strength of that illusion differently, the situational factors you're talking about. And to cut a long story short, essentially the argument goes something like, if you imagine this thing with these two arrowheads, and now turn it, and now look at the corner of the room looks rather like a vertical line with two arrowheads going at the side. And the argument goes, if you grow up in a Western society full of houses made like boxes, you see a lot of lines like that. Whereas if you grow up on the plains or in a Maasai hut, which is circular, you don't see a lot of things like that. And that modifies your visual cortex architecture, and that in turn creates a cultural difference in the strength of an illusion. It's a good story, really good story. I really like the story, and of course it's a great experiment. I think at the moment we just don't have the data to answer that. If the general question is, do we imagine that the context in which you grow up influences individual differences, the answer has to be yes. I think the outstanding question is, for the types of individual differences I've shown you, say early visual perception versus metacognition, do we think the relative influence of cultural or situational factors versus innate differs? I think the answer to that would also probably be yes. So for example, early visual cortex, the architecture is laid down by and large prenatally, it's myelinated at birth. And so I would imagine that the plasticity of primary visual cortex to cultural influences is rather different to that for, say, prefrontal cortex. But ultimately, it's an empirical question. More research is needed. Any more for any more? You're sitting quietly. Go for it.
Sorry, I didn't want to.
No, you should always ask questions.
I guess this is a bit more philosophical because I'm not a psychology student.
So even better.
Well, when you talked, I thought the most interesting part was about introspection bit. And
Yeah.
Parietal cortex. And you said that basically the size of the parietal cortex influences how good you are in introspecting yourself.
Okay, yes, sort of. I showed you the size of that frontal bit of the brain.
Yeah.
I didn't say determine, so that goes back to the question we had about developmental. It correlates. So it could be that you're really good at introspecting, and that makes that bit of the brain change in structure. Or it could be that that bit of the brain is different in structure and makes you good or less good at introspecting. That's important. I mean, maybe the study I didn't show you was the Politics in the Brain study, which you can all look up, which I did at the behest of Radio 4, where Ryota and myself measured the political preferences. Hey, it's almost a general election. Let's talk about that for 2 minutes. You measure, it turns out political scientists say that there's one cross-cultural scale of political attitudes, which basically goes from liberal to conservative on a 5-point scale, right? You're either very, very conservative, in the middle, or very, very liberal. right? And apparently all cultures can place themselves on that. If you look at variability in a population like this, you turn up with two brain structures that survive and replicate with a relationship between structure. The anterior cingulate, which as somebody you know is involved in error detection and uncertainty, is thicker, or it has more grey matter in it for those who are more liberal, who describe themselves like that. Whereas the amygdala, the structures you know involved in emotional perception and fear perception and all sorts of emotions, in fact, is larger or has more grey matter if you describe yourself as conservative compared to liberal. Now that, apart from the fact that again shows you can take a complex behaviour and show a relatively simple and replicable result, illustrates the causal arrow is not clear there, right? So do we believe that because your amygdala is bigger, you vote conservative? I don't think so. But equally, why do you vote conservative? And why, for example, are voting preferences heritable? So people have studied that in twin studies and shown the heritability is about 0.6, which is quite scary, I think. You know, so there is an innate component as well, but it just makes clear the sort of nature-nurture point. Right, sorry, I just riffed on something I wanted to tell them about while you were thinking about the rest of your question.
No, you gave me the answer because, like, I thought that then I was about to ask, like, is that then a matter of luck if you're good at self-reflecting because you got a bigger part of the frontal brain, or
yeah.
But we don't know.
And I think that's the really interesting question, isn't it? I mean, causal questions are always more interesting than correlational ones, and they're always harder to look at. So the answer to the developmental question illustrates that you can use time to help answer those questions, but that involves you studying people for a long period of time. You can use the disruption to answer a simple causal question, which is this area really involved or just sort of epiphenomenally involved? That was the parietal cortex answer. But if you want to know how do these come about, which I think is the natural next question, that's altogether harder. It's the different level of difficulty in terms of scale and longitudinalness.
I think we're out of time, but if you guys have more questions, we will be in the Wilson Common Room in about 10-15 minutes.
Yep.
And there will be wine. Come join us and the speaker. You can listen to the discussion, ask more questions. Have a great.
Thank you very much.